Hello everyone, myself Guruvinder Kaur. Today I am going to start with the Biochemistry BP 205T Unit 5 Enzyme. Here in this unit enzyme we will cover introduction to the enzyme, enzyme nomenclature and classification, mechanism of action, how the enzyme work in the body and what are the factors which affect the enzyme activity. The next we will cover enzyme kinetics, then enzyme inhibitors, enzyme regulation, isoenzyme, enzyme, and last we will discuss about the therapeutic and diagnostic application of the enzyme. So, now we are going to start one by one. In this lecture we will cover the introduction of the enzyme and enzyme nomenclature and classification. Introduction of enzyme. The first thing is what are enzymes? So, the enzymes are the organic macromolecule synthesized in the living cells, increases the rate of biological reaction without being changed in the overall process. Now, the term here is macromolecule. The macromolecule is a large grand molecule, bada molecule which is inside the body, which is synthesized in the living cells. जैसे carbohydrate, lipids, proteins, these are the macromolecules, but the इनकी जो मोनोमर यूनिट्स है, जैसे कार्बोहाइड्रेट की मोनोमर यूनिट्स होती है ग्लूकोज, दिस इज द माइक्रोमोलिक्यूल. प्रोटीन की मोनोमर यूनिट है अमीनो एसिड्स, दिस इज द माइक्रोमोलिक्यूल. डीएनए एंड आरएनए इज द मैक्रोमोलिक्यूल, न्यूक्लियोटाइड इज द micromolecule. So, the enzymes are the organic macromolecules synthesized in the living cells, increase the rate of biological reaction or biochemical reaction without being changed in the overall reaction. The enzyme increases the rate of reaction by decreasing the free energy barrier that separates the substrate or we can say reactant. In the non-living cells where the catalyst is used, we use the reactant for the reaction, but in the living cells where the enzymes are using, where the enzymes are acting as a catalyst, we will use a term substrate for the reactant. Now, enzyme catalyze the reaction, enzyme catalytic reaction जो होती है that occur generally at the normal body temperature, atmospheric pressure, nearly neutral pH and have higher rate of reaction as compared to the catalyst. Now, coming to the nature of the enzymes. Enzymes are biological catalyst. It means they are present in the biological system. They catalyze the biochemical reaction. They are colloidal in nature. Colloidal nature means because why they are colloidal in nature? Because the enzymes are very large molecule, Gantt size molecule. If we see the molecular weight of the enzyme, that is almost 12,000 Dalton to 1 million Dalton. So, it is a very large enzyme. That is the reason why the enzymes are due to this large size of the enzyme molecule possess extremely low rate of diffusion. They diffuse in the very low रेट, बहुत ही धीरे धीरे रेट से वो डिफ्यूज होते हैं एंड दे फॉर्म अ कोलाइडल सिस्टम. छोटे छोटे पार्टिकल्स एग्रीगेट होकर कोलाइडल सिस्टम बना लेते हैं इन द वाटर, इन द वाटर सिस्टम. द कोलाइडल नेचर होने की वजह से, ड्यू टू द कोलाइडल नेचर ऑफ द एंजाइम, दे आर नॉन डिसोशिएबल, बट दे आर सर्टेन एंजाइम विच आर डिसोशिएबल ड्यू टू द प्रेजेंस ऑफ सर्टेन कोफैक्टर और कोएंजाइम्स. But the enzymes which are not having any coenzyme, they are non-dissociable. The next property is thermolabile. Thermolabile, it means they can destroy, the enzymes are destroyed or deactivated by the heat. So, they are active at the optimum temperature only. They have the maximum activity at the maximum temperature. The next property is the protein in nature except the ribozyme. All the enzymes are proteinous in the nature. सारे enzyme protein के होते हैं, but remember all the proteins are not enzyme. Here is the exception, that is riboenzyme. Riboenzyme क्या होते हैं? These are the RNA which are the type of RNA which catalyze the reaction. ये एक ऐसे RNA का टाइप है जो कैटालिस्ट का काम करते हैं, that is riboenzyme. प्रोटीन होने की वजह से दे में बी मोनोमेरिक और ओलिगोमेरिक. मोनोमेरिक सिंगल पॉलीपेप्टाइड चेन के बने हुए होंगे या फिर ओलिगोमेरिक यानी कि 2 और मोर देन 2 पॉलीपेप्टाइड चेन्स के बने होंगे. The next one is highly specific. These are very specific in nature. एक particular enzyme particular reaction के लिए ही use होगा. For an example, if you're talking about the glucokinase, तो glucokinase क्या होगा? Glucose को convert करेगा glucose 6-phosphate में. If you remember the cycle metabolism of carbohydrate, glycolysis, तो आपको glycolysis याद होगा 10-step process होती है जहाँ पर first step glucose is converted into a glucose 6-phosphate. The enzyme is acting as a hexokinase or glucokinase. तो means ग्लूकोज को ग्लूकोज 6-phosphate में कन्वर्ट करने के लिए एक ही तरीके का एंजाइम यूज हो रहा है. इसी तरीके से बहुत सारे एग्जांपल्स हैं. तो एक एंजाइम एक ही तरह के रिएक्शन पर्टिकुलर एक रिएक्शन के लिए ही होता है. सो इट इज वेरी स्पेसिफिक. सम एंजाइम रिक्वायर्स एन एडिशनल नॉन प्रोटीन कंपोनेंट फॉर इट्स ऑप्टिमम एक्टिविटी. दिस एडिशनल नॉन प्रोटीन कंपोनेंट इज कॉल्ड एज cofactor, right? These may be loosely bound or tightly bound to the protein portion of the enzyme with the help of a covalent bond. या तो tightly bond होंगे या तो loosely bond होंगे. जब ये non protein part जो है, if this non protein part of the enzyme is inorganic, then it is called as activators, or sometimes it is called as cofactor. Also in certain book it is written as a cofactor or activator. And but when this is organic in nature, this non-protein part in the enzyme is organic in nature, then it is called as coenzyme. Enzyme without any cofactor, it is referred as an apoenzyme. The complete catalytic activity of enzyme is called as holoenzyme. It means the holoenzyme is the active enzyme which is made up of 2 parts, that is apoenzyme, a protein part, and the coenzyme, a non-protein part. Okay, the enzyme is made up of 2 parts, apoenzyme, non-protein, and the coenzyme, a non-protein part, and apoenzyme, a protein part, which is combinedly called as a holoenzyme, which is the active enzyme. The another term always we use is the prosthetic group. It is used when the non-protein moiety is tightly bond or covalently bond. tightly bond होगा तो obviously covalent bond होगा with the apoprotein. यानी कि जब coenzyme इस apoprotein से covalently bond करेगा, एक strong bond बना लेगा, तो उसको हम prosthetic group के नाम से represent करेंगे. कुछ इस तरीके से you can see in the figure. Here we have a apoenzyme, that is a protein portion which is inactive. When a cofactor, that is a non-protein part, come with contact with the apoenzyme, then it form a active part. Here it is the substrate where it will combine and give the catalytic reaction. Similarly, here also a cofactor is coming and combining with the apoenzyme, the protein part. The apoenzyme become active by the binding of this cofactor or coenzyme. एक्टिवेशन होगा और यहाँ पर ये जो सबस्ट्रेट है इससे इजीली फॉर्म हो जाएगा. द holoenzyme is formed when association with the cofactor or coenzyme binds with the enzyme active site. Let us take some examples of cofactor. Zinc कार्बोनिक एनहाइड्रेस पे, जिंक एल्कोहलिक डिहाइड्रोजिनेज एंजाइम्स के लिए, Fe3+ or Fe2 ferric or ferrous ion for the cytochrome or hemoglobin, also for the pyridoxine, copper 2+ or copper plus cytochrome oxidase, and calcium, potassium and magnesium ion for the pyruvate phosphokinase. Let us see the example of coenzyme. We have vitamins which will act as an coenzyme जिनके नाम इस तरीके से and the function. For an example, B1 thymine pyrophosphate, that is TPP. The function is क्या करता है ये function क्या करेगा, decarboxylation की reaction को enhance करेगा, decarboxylation reaction में participate करेगा. The another one is B2 riboflavin and FAD, that is flavin adenine dinucleotide, hydrogen transfer का काम करता है. B3 niacin nicotinamide adenine dinucleotide NAD प्लस हाइड्रोजन ट्रांसफर का काम करेगा. इसी तरीके से B4 टेट्राहाइड्रोफोलिक एसिड फॉर्मुलेशन का काम करता है. B5 कोएंजाइम A ट्रांसफर ऑफ एसाइल ग्रुप का काम करेगा. B6 पिडॉक्सिल फॉस्फेट ट्रांसएमाइनेज का काम करेगा. यूरिया साइकिल का आपको याद हो तो कोबाल्ट B12 दैट इज कोबालामाइड एंड इट विल ट्रांसफर अ कार्बोनिल ग्रुप H biotin and it will do the carboxylation. Moving forward, the next term which we have to understand, that is active site. What is active site? The active site is the enzyme, the active site of an enzyme is the region where the substrate or the cofactor or the prosthetic group combine, bind करता है, वो form, वो पार्ट जो है कहलाएगा आपका एक्टिव साइट. अगेन रिपीटिंग, द एक्टिव साइट ऑफ एन एंजाइम इज द रीजन दैट बाइंड सबस्ट्रेट और कोफैक्टर एंड प्रोस्थेटिक ग्रुप एंड कंटेन रेसिड्यू दैट हेल्प टू होल्ड द सबस्ट्रेट. सबस्ट्रेट को होल्ड करने का काम करेगा. यू कैन सी हियर दिस इज द एंजाइम. दीज लार्ज ग्रुप्स आर द बाइंडिंग साइट, होल्ड द सबस्ट्रेट इन द प्लेस. This is the small little groove which is called as a catalytic site where the reaction will take place, and this is the substrate. This is called as active site. The active site generally occupy less than 5% of the total surface area of the enzyme. Active site has specific shape due to the tertiary nature of the protein, या tertiary structure of the protein. A shape in the change in the shape of the protein affect the shape of the active site and the function of the enzyme changes. The function of the enzyme will change. Now coming to the type of enzymes. So we have two type of enzyme. First one is intracellular and extracellular. Intracellular नाम से ही पता चल रहा है, इंट्रा यानी कि सेल के अंदर, ठीक है. तो इंट्रासेलुलर और जाइमेज, these type of enzyme, these intracellular enzymes are synthesized and retained in the cell and used by the cell itself. It means जिस सेल पे ये synthesize हो रहे हैं उसी सेल में ये रहते हैं, उसी सेल पे ही काम करते हैं और वहीं destroy हो जाते हैं, right? For an example, they are found in cytoplasm, nucleus, mitochondria and cytoplasm. Example इसका है oxidoreductase, catalyze the biological oxidation reaction. The another one is extracellular or zymogen or proenzyme. नाम से extracellular, it means these are the enzymes which are synthesized in the cell but excreted from the cell and work externally. यानी कि जहाँ पर ये सेल सिंथेसाइज हो रहे हैं, जिस सेल के अंदर these enzymes are synthesized, they are not working in that particular cell but they are excreted out and move somewhere else and act somewhere else. The number of, बहुत सारे देखें तो number of the proteolytic enzymes found in the blood or in the digestive tract are present in an inactive form, or we can say precursor form. For example, chymotrypsin, it is secreted by pancreas as a chymotrypsinogen. It is activated in the digestive tract by the proteolytic enzyme trypsin. Now coming to the nomenclature and classification. Traditionally, enzyme often were named by adding suffix "ase" to the substrate upon which they act. इतने सारे millions of enzymes होते थे, तो traditionally हम इसको indicate करते थे कि जिस सबस्ट्रेट पे ये एक्ट कर रहा है उस सबस्ट्रेट पे हम एक सफिक्स स्टेम लगा देते थे एज, उसके नाम पे नाम दे दिया जाता था कि ये पर्टिकुलर एंजाइम है। जैसे लाइपेज, लाइपेज एक्ट करता है लिपिड, तो लिपिड पे लिपिड का आई पी लिया और सफिक्स एज लगा के लाइपेज बना दिया गया। यूरेज एक्ट एज अ यूरिक एसिड, न्यूक्लियेज एक्ट ऑन द न्यूक्लिक एसिड। इस तरीके से ट्रेडिशनली एंजाइम्स को नाम दिए जाते थे, बट इसकी वजह से क्या होता था, बहुत कंफ्यूजन हो जाता था इस ट्राइबल नेम की वजह से। तो we were not able to explain कि what type of reaction is going on, कौन सा cofactor लग रहा है, कहाँ particularly ये reaction हो रही है. So there was too much of confusion in this tribal name. To overcome this, the new system of the nomenclature of the enzyme was developed in the 1964 by the Enzyme Commission of the International Union of Biochemistry and Molecular Biology, that is IUBMB, or simply it is called as IUB, or the International Union of the Biochemistry. According to that, all enzymes are assigned as a EC number. इनके अकॉर्डिंग जितने भी एंजाइम्स हैं उनको EC का नंबर दे दिया गया. The classification does not take into account the amino acid sequence, protein structure or the chemical mechanism. ये जो nomenclature है ये आपको नहीं बताएगा कि ये जो एंजाइम है वो कौन से अमीनो एसिड से बना हुआ, बिकॉज द एंजाइम्स आर मेड अप ऑफ प्रोटीन. प्रोटीन किससे बने होते हैं, अमीनो एसिड्स के बने होते हैं. तो ये नॉमेनक्लेचर आपको ये नहीं बता सकता कि किससे क्या होमोलॉजी है या कौन से सीक्वेंसेस से, किस तरीके के सीक्वेंसेस से एंजाइम का फॉर्मेशन हुआ है, इसका पूरा प्रोटीन स्ट्रक्चर क्या है या केमिकल मैकेनिज्म क्या है, ये नहीं बताएगा. बट दे विल टेल कि व्हाट टाइप ऑफ रिएक्शन इट इज डूइंग. The EC number had 4-digit numbers. For an example, A, B, C, D, where A is the class, B is the subclass, C is the subclass, and D is the subclass. The B and C digit describe the reaction, while the D digit is used to between the different enzyme of the same function based on the actual substrate in the reaction. The enzyme are divided into 6 major class. Each class on its own represent the general type of reaction through about by the reaction of that class. First one is EC1, that is oxidoreductase. The enzyme involved, the name as it is saying, oxidoreductase. It means the enzyme involve the transfer of hydrogen or oxygen atom or the electron from one substrate to the another substrate. Okay. It means the oxidation and reduction reaction is taking place. It is also called as oxidase, dehydrogenase or reductase. Note, जो याद रखने वाली बात है, that since these are the redox reaction, an electron donor or the acceptor is also required to complete the reaction. Okay, since this is the redox reaction, an electron donor as well as acceptor, donor या फिर acceptor is also required to complete the reaction. For an example, we have phosphate dehydrogenase. कैसे होगा रिएक्शन ऑक्सीडोरेडक्टेज का, एक का ऑक्सीडेशन होगा दूसरा रिड्यूस हो जाएगा. इफ यू सी AH2 एज एन एग्जाम्पल, B, it will give B and B plus BH2. The another class is EC2, that is transferase. As name indicates, transferase यानी कि ट्रांसफर कर रहा है. ट्रांसफरेज, ट्रांसफर किसका ट्रांसफर कर रहा है, फंक्शनल ग्रुप का. The transferase enzyme do the function of transferring the functional group. For an example, you can see from this reaction we have AX. X अगर कोई functional group है, यहाँ पर B act हो रहा है तो functional group का transfer हो रहा है B to F, BX का formation A. For an example, methyl transferase and carboxy transferase. The next one is EC3, that is hydrolase. Name indicate hydrolase, hydrolytic reaction. These hydrolase catalyze the hydrolytic reaction. It include lipase, esterase, nitrase, peptidase, protease, etc. कैसे? AX is the substrate where the H2 molecule will act. क्या करेगा, XOA hydrolysis and HA will be formed. For an example, carboxylic ester hydrolase, glucose 6 phosphate are the example. Next one is EC4 lyase. Enzyme involves non-hydrolytic, right? The enzyme involves non-hydrolytically removal of a functional group from substrate, creating a double bond in the product, or the reverse reaction, that is addition of a functional group across the double bond. ये जो EC3 था आपका, EC3 की तरह एक्ट नहीं करते हैं, हाइड्रोलिसिस कराता है, बट नॉन हाइड्रोलिटिक रिएक्शन है. दे विल बी अ रिमूवल ऑफ अ फंक्शनल ग्रुप बाय द फॉर्मेशन ऑफ अ न्यू डबल बॉन्ड बिटवीन दैट, ठीक, और create a double bond in the product or reverse the reaction, that is addition of the functional group across the double bond. Includes decarboxylase and aldolase in the removal direction and synthetase in the additional direction, right? Includes decarboxylase and aldolase in the removal direction and the synthetase in the additional reaction. You can see A and B is the compound when react with XY, right? Then it form AX and BY. The next one is EC5 isomerase. These isomerase catalyze the isomeric reaction, isomerization of the reaction. They catalyze the isomerization of the reaction. A is converted into a different A, right? Isomer के form में convert करता है. Epimerase, racemase are the example. EC6, that is ligase, catalyze the synthesis of the various bonds, especially that is mostly that is CX bond, coupled with the breakdown of the energy containing substrate. For example, the formation of ATP cleavage, formation of bond with the ATP cleavage, formation of bond with the ATP cleavage. B plus A, there will be the formation of AB compound, addition हो रहा है A and B का with the help of the energy in the form of ATP. So, the ligase catalyze the synthesis of the various bond coupled with the breakdown of the energy containing substances. Energy containing substances क्या हो सकते हैं बायोलॉजिकल सिस्टम के अंदर? ATP हो सकता है, AMP हो सकता है, GTP हो सकता है, एनी एनर्जी कंटेनिंग मॉलिक्यूल होना चाहिए दो सबस्ट्रेट को बाउंड करके एक सब्सटेंस बनाने के लिए. These are the summary I have given here. ऑक्सीडोरिडक्टेस काम क्या करता है? इलेक्ट्रॉन ट्रांसफर का. फॉर एग्जाम्पल अल्कोहल डिहाइड्रोजिनेस. ट्रांसफरेज क्या काम करेगा? ट्रांसफर ऑफ द कार्बन, नाइट्रोजन और फॉस्फोरस कंटेनिंग ग्रुप. फॉर एग्जाम्पल हेक्सोकाइनेज. हाइड्रोलेज, बॉन्ड क्लीविंग बाय द एडिशन ऑफ द वाटर. ट्रिप्सिन एग्जाम्पल. लाइज, cleavage of the CC bond, CS bond or CN bond often to form a double bond is such like this. That is the, for an example we have pyruvate decarboxylase. The another one EC5 का था. EC5 it is isomerase, that is the formation of a optical or geometrical isomerase जो isomerism कर रहा है. epimerism इसका example था. Biological system में इसका example देखेंगे. We have a malate isomer. And the last one EC, this is ligase, the hydrolysis of high energy phosphate to form a new bond, that is pyruvate carboxylase. Hope you understand this. Thank you.